Good morning, everybody. It's my pleasure to chair today seminars given by Dr. Mohit Kumar. He's recently joined our group in the molecular bionics lab as a junior Kaisha junior leader fellow, and he's brought some very interesting expertise into our group. And he will tell us all about today how he came about to start working on auto-equilibrium materials and lifelike structures. Mohit, I'm not going to say much more. I'm going to leave everything to you to explain.
Sure. Okay. Thank you, Pepe. Good morning, everybody. It's a pleasure for me to be here today talking to you all about my research that I've been doing for some time. Before I start, I would probably give you a brief introduction about myself and where I come from and my kind of research, and then get into the work that I have done. So I am, okay, if the slide moves, I would, but looks like it doesn't want to move. So let's try something else. I was told that the slide freezes once in a while, so I will try to reshare the screen and see if it works. No. Okay. I hope it moves now. Yeah, okay. Okay, it's moving. That's a good thing. You can see my new slide, right? Anyone?
Yes, yes.
Okay, cool. Thank you. Yeah, so to start with, I did my PhD in India from Bangalore, and my training has mostly been as a chemist. So I did my master's in chemistry, and then my PhD was based on studying soft material made with small molecules, organic material, and its function. And after my PhD, I moved to New York for my postdoctoral study at City University of New York, and that's where I was there for last 5 years before I moved to IBEC in Barcelona after having received the La Caixa Junior Leader Fellowship. So most of my work that I would speak today will be mainly what I did in New York, and since I joined here in June, I would really not give you many results from here, but just to give you an idea about what I plan to do here, because the results are not yet ready to be presented, I think. So with that, let me start. Here is the outline of my presentation today. I would first introduce you to the idea of soft matter and supramolecular polymers, and then show how these can be used to form nanomaterials with lifelike function. And I would define what I mean by lifelike function and show its three property in terms of having a temporal control over structure and its function. And finally, you visualize them with super resolution imaging. And at the end, I will give you an idea about what I plan to do in future in IBEC before I conclude. So to start with soft matter, I'm sure this slide, everything presented here is you are aware of in some form of or other. These are various examples of soft matter and many more. But one thing that you would see in common in all of these are that they are made with polymers, conventional polymers, where monomers are held together by covalent bond. I see a background noise. I don't know if it is from my side or somebody else's mic is on.
I don't hear any background noise, to be honest with you.
Okay, yeah, now it's clear. Thank you. Thank you. Okay, so and this has been used in food industry, cosmetics, and personal care, etc. But these, to be highlighted, these are materials made with polymers where monomers are held together by covalent bond. And what I work with are so-called supramolecular polymers. In a conventional polymers, the monomers are held together by covalent bond, and therefore they are rigid bond. Here, monomers are held together by weak non-covalent interaction. They can be hydrogen bonding, aromatic interaction, sulfophobic interaction, van der Waals, etc. But these are weak non-covalent interaction. These are not by covalent bonding. So with this, what you gain is you gain an extra level of dynamicity into the system, and that results in various properties and function. But then one question can arise as to if these are really held by such non-covalent interactions which are weak, then the whole structure itself should become very weak. Now, just to show you that these non-covalent interaction can also result in very much like polymeric material, which are solid polymers. And again, to highlight here, the monomers are small molecules which are held together by weak non-covalent interaction. So it can be highly dynamic, but it can also be stable, as you can see here. So having shown you these supramolecular polymers, what all has been done with such a field. Now, at this point, we can make variety of structures from these supramolecular polymers, wherein monomers are held by weak interactions. They can form fibers, tapes, tubes, or variety of other structures like sheets, etc. And they have become now a very, very new field of research for, I would say, last 30 years or so. And for a variety of applications in the field of organic electronics, light harvesting, or for interfacing with biology in terms of cell culture medium, or for interfacing with neuron cells, or for nanomedicine, etc., etc. So just to say that these supramolecular polymers now has been used for various function applications. But here is like I would say my understanding of how soft material has evolved over time. So we initially started with polymeric structures, which have revolutionized itself our life. But they are passive matter. So once they are formed, nothing happens to them. They don't change. They don't respond to anything. Then we started to move towards from passive matter to dynamic matter or responsive matter, like liquid crystal structure, the lipid vesicles, the pH responsive structure. So these are responsive material which respond to external stimuli. So these are they can be either pH, light, etc. But if nature was to be the inspiration for such a design, you can see that the complexity is much more higher, and it's much more complex in nature, wherein nature uses active and adaptive material which can adapt to external stimuli, and they consume chemical fuel to give you various function. So this is where my research would progress towards, from dynamic and responsive matter to active and lifelike matter. Now, what do I use to make such material? So as I said, I'm a chemist, so I make variety of molecules. These are synthetic molecules. But I also use variety of biological molecules to incorporate into the synthetic structure. So I use various amino acids or nucleotides, enzyme, and various biological machinery like metabolic system, etc. So I combine the two fields of synthetic chemistry as well as biomolecules to bring this lifelike function into the material. Now, when I talk about lifelike material, what I mean. So let me give you what I intend to mean by this. Now, clearly, these two are very different. The one on the left is a synthetic plastic material, wherein on the right is a living plant. Now, the difference in these two would become much more evident if you look close into it. So if you look at synthetic plastics, They are made with molecules which somehow forms a material. And once it forms a material, it exists as it is in its thermodynamic equilibrium. The idea being, if you keep this plastic in a box, it would still exist as it is. But if you try to keep the same thing for a plant, the plant cannot survive in a closed box. It needs to interact with its surroundings. It needs to exchange energy and matter from its surroundings. So basically, it needs some kind of a chemical energy for its existence. And what does it do with these chemical energy? One of the many things that it does is that it can adapt to the surroundings. And here the structures form and degrade over time, and they form with spatial resolution and temporal resolution. So for example, cell division itself requires a lot of chemical energy, and then it can divide into two daughter cells. So this is done with very spatial precision and temporal control. So that's the beauty of having structures which are which has a life of its own. Now, if you think of synthetic materials that we make in our lab, or any material that we usually work with, for example, a pen or a cup, etc., everything we make exists as it is forever. It doesn't require any chemical energy for existence, except for biological systems like plants. So our idea is, can you make materials which have a lifetime of its own? So material which exist for a certain period of time. Now, what has been done in literature in this field? So people in the researchers in the last 10 years have shown that now structures can be made which have a lifetime of its own. So you can see, for example, here you have a solution which over time becomes a gel, which indicates that the self-assembled fibers have formed over 1 hour. But then over time, this fiber again break down into monomers to give you this solution again. And you can see here that the fibers are beginning to form into longer and longer fibers, and some are also degrading. So this is like creating transient nanofiber, or fibers which have a lifetime of its own. And here on the right is actually microtubule structure, which can walk and move around. So it's also driven by some kind of a chemical energy into the system. So when we look more closely, what we find is that there are two competing reactions. A first chemical reaction which leads to formation of these structures, and a slow degradation reaction. And if the rate of formation is higher than the rate of degradation, you would have the self-assembled structure existing in the system, depending upon the chemical kinetics. So to say that transient structures have been reported, or structures which have a lifetime of its own. What was not reported is, if these structures also had some function, you can control the lifetime of the function. And that's exactly what biology does. When it tries to create a temporal control over its structure, it tries to also control the function for a given period of time. That's when my work came into picture. I was interested in saying, if I can control the structure for a given time, and if this structure does a function, I should be able to control the lifetime of the function as well. So with that idea in mind, I designed this molecule, which is a naphthalene diamide-based system. So this molecule is an organic semiconductor, so it can conduct electricity. And interestingly, on either side of this molecule, it has tyrosine methyl ester. Now, this is an amino acid with a difference that you have an L enantiomer on one side and D enantiomer on the other side. So the molecule has a plane of symmetry in the middle. It's a meso molecule. It's achiral for all practical purpose. But remember, I was telling you that you need a fast reaction and a slow reaction for such transient structures to form. So if I do an enzymatic reaction on this molecule, like a protease reaction, reacting to this molecule, protease has a preference to react with the L enantiomer. So there would be a reaction happening at the L terminal that will lead to a break in symmetry of the molecule, and you would see a chiral self-assembly. But it can also slowly react with the D terminal, and thereby again reverting back to its symmetry. So that would be the slow reaction. You have a fast reaction at the L terminal and a slow reaction at the D terminal. Now, what kind of reaction I'm talking about? I'm talking about this molecule reacting with one of these amino acids, either hydrophilic amino acids or hydrophobic amino acids, in presence of an enzyme alpha-chymotrypsin, which is a protease. So what we found is that, as expected, reaction will happen fast at the L terminal. So over 3 hours, reaction happens. But this reaction, the pathway of chemical reaction, is controlled by the type of amino acid we put into the system. So if you put hydrophobic amino acid like aspartic acid or glutamic acid, what we see is that hydrolysis happens at the L terminal to give you an acid over 3 hours. But then if you wait for long enough time, like 2 weeks, that reaction happens also at the D terminal to give you a diacid. Wherein with hydrophobic amino acid like leucine or phenylalanine, you see a dipeptide forming at the L terminal. And this over time, when reaction happens at the D terminal as well, this would happen in like 2 weeks time. You see that hydrolysis happens at the D terminal. So you can see that the chemical pathways can also be now controlled by the type of amino acid that you have put into the system. So this control over chemical pathway is also affecting its supramolecular structures over time. So here I would like to introduce to you the idea of circular dichroism and left and right-handed helical assembly. Now, circular dichroism, you might be aware of, has been used for characterizing various proteins. But with aromatic molecules and the chromophores, it is used to characterize the organization of these molecules into either a left-handed helix or a right-handed helix, which is dependent upon the direction of circular dichroism signal, whether you get a positive signal or a negative signal. So now if I were to monitor the circular dichroism of this molecule in presence of various amino acids and chymotrypsin over time, what we see is very interesting. So if you focus on the red curve here, you see that initially the CD signal is 0, indicating it's an achiral molecule, which is expected. But over time, the circular dichroism signal starts to increase in the negative direction, which indicates a helical organization of these molecules. And this happens over like 2-3 hours. But if you wait for long enough time, like 2 weeks time, you see that the CD signal starts to decrease and goes back to 0. So effectively what has happened is you have created a transient chiral fiber or transient chiral structure. So we start with a achiral molecule, achiral structure. Over time, it becomes chiral of one particular helicity. And then if you wait for long enough time, the system will evolve into an achiral structure again. So that's like creating a temporal control over the chiral structure. Interesting, if you just change the amino acid from hydrophilic like aspartic acid to leucine, you get a completely different supramolecular trajectory. Now you go from achiral structure over 3 hours to a right-handed helical structure. And this chirality remains permanently. So it's a delayed chirality, but permanent. And then intermediate polarity like serine gives you an intermediate behavior, and leucine and tyrosine remains completely achiral. So what you see is that amino acid sequence has a very important role to play in deciding the supramolecular trajectory of these structures. So here you can clearly see that the two pattern emerges. One is the red, which is a transient chiral structure, and the other one is yellow, which is a delayed but permanent chiral structure. So if you look more close into what is happening, what we see is that initially, when the reaction happens at the L terminal, it forms a dimeric structure with glutamic acid, and this is an amphiphile, wherein on the D terminal is hydrophobic, on the L terminal becomes hydrophilic. That self-assembles into a chiral structure, and it gives you a strong CD signal, which you can see here. But over time, when you wait for long enough, over 2 weeks actually, the hydrolysis happens at the D terminal, and that gives you an acid. Now it's a diacid, so acid on both sides. This makes the molecule extremely hydrophilic, and therefore the whole supramolecular structure collapses, and you have only globular aggregates left. And that's why you see that the circular dichroism signal goes back to 0. So when we look at the TEM, you see that the two-dimensional platelet-like structure, it forms one-dimensional fibers, which are chiral. And over time, over 2 weeks time, it forms globular aggregate. So it's like creating transient helical fibers, wherein the helical fiber have a lifetime of its own. But when we switch the amino acid to leucine now, you see a very different behavior, wherein it starts with forming a dipeptide at the L terminal, but this doesn't self-assemble into a chiral structure because it's hydrophobic on both sides. When reaction happens at the D terminal, it forms an acid, and that becomes hydrophilic. The whole thing is an amphiphile now, and that becomes that forms a chiral structure. So you can see that the CD signal here goes from 0 to 0 again till 3 hours, and then starts to increase in the right direction. And TEM shows that platelets goes to one-dimensional fibers, but these fibers are not chiral anymore. The chirality comes when it switches from the fibers to tubular aggregates. And these tubular structures, you can see some of the openings of the tubes as well here in these micrographs. That these tubular structures are the ones which are responsible for the chirality. So with that, here I want to show that as it is so important that a very simple amino acid can also control the supramolecular trajectory of these structures, where it goes from either a transient chiral nanofiber to a permanent chiral tubular structure. So when we did this detailed analysis of this, what we confirmed is that depending upon the amino acid you put into the system, you position these amino acids either into the core of the structure or at the periphery. And depending upon the position of these amino acids within these nanostructure, you decide the helicity as well as the lifetime of these structures. So now coming to the main motivation that we started with is, remember we have an organic semiconductor embedded within these structures. So when we start from platelet to one-dimensional fibers, as these fibers form, these fibers can start to conduct electricity. So it's in some way like a nanowire in the system that we create. But then these structures also break down eventually, and therefore the electrical conductivity should also decrease. So when we probe the electrical conductivity of such system over time, you see that there is an enhancement in current, which indicates that as fibers are forming, they are also starting to conduct electricity. But over time, as the structures start to degrade, you also see that the electrical conductivity also decreases and comes back to where it was before. So this clearly indicates that we have created a transient electroconducting structure. Now, as you imagine that most of the circuits that we work with are all stationary. That is, once you have the electrical circuit, it remains as it is. But the neural networks are formed on demand, and it changes its orientation and connections. Here, in these synthetic structures, what we showed is that now you can make these fibers which can conduct electricity for a certain period of time. So the idea was that if you want to really electrostimulate, let's, for example, electrostimulate a neuronal cells, you can electrostimulate with these structures over which, over time, once the stimulation part is over, it will degrade and then clear out from the system very easily. So this is the first example where we showed that transient conductivity can be controlled over time in these soft structures. But the limitation of the system was that the lifetime was not under our control. It was in few hours to 2 weeks time, and we wanted to make it much shorter and better controlled. So for that, I had designed a new system, which is made up of, again, tyrosine methyl ester derivative. But this does not have the D enantiomer, so it's completely an L enantiomer. So the rate of reaction becomes much faster, and now you can control the lifetime of these structures. So this forms a dipeptide in presence of an amino acid and an enzyme. And this dipeptide self-assembles into a nanofiber. And over time, the dipeptide is hydrolyzed by the same enzyme to give you an acid derivative, and that's where the disassembly happens. So you can see that we start with a solution which forms a gel over time, and then it degrades back into solution again. Now here what we found is that depending upon the hydrophobicity of amino acid, which also dictates the self-assembly propensity of these structures. Now you can control the lifetime of these materials from few minutes to few hours to even days time scale. So remember, one of the feature of living structure or living material, living entity is their lifetime itself, wherein materials that we usually work with do not come with their own lifetime. I know there is an expiry date, but that does not mean that the structure is broken down. Here is a system wherein you can now dial in how long a self-assembled structure should remain into the system, and therefore how long the function of these structures should remain into the system. So again, we did microscopic studies to find that these are indeed one-dimensional fibers that exist for a certain period of time before they break down into random aggregates. And this also had a dye incorporated, so we could visualize them with confocal microscopy. And you can see that one-dimensional fibers in solution. But as you notice here, these are various snapshots at various time. They are not real-time images. The limitation of such a system was that we could only do this at various time. If you want to visualize structures in real time, we should be able to image them in solution, like in confocal, but with much better resolution than a confocal, to be able to see how the supramolecular transformation happens from a random aggregate to a one-dimensional fiber before it degrades. So to do that, we have to move away from conventional confocal microscopy into something more high resolution. So here I show you that the confocal microscopy has a resolution limit, I would say, of 200 nanometers. And the other limitation of such microscopy is that you need to covalently link a dye onto the molecule, a fluorescent dye, before you can image them. So covalent linkage is tedious, it's expensive, and it can also affect the self-assembly process itself, because the molecule is covalently attached to the dye is covalently attached to the molecule. So it is then we decided to use a super resolution microscopy, which is stimulated emission depletion based microscope. It's called STED in short. Now, STED is we chose this STED first because this was the instrument which we bought in the department. But we bought this also because of the advantage that here you can image these structures in situ. So you don't need to do a sample processing and changing the solvent. You can do in whichever solvent you have been working. And it can do fast enough imaging for you to visualize or capture a real-time video of these structures. And then the second idea was, can we also simplify the process of covalent linking of dye onto the molecule? So covalent linking, as I said, is tedious and requires it requires a lot of effort to make it. My idea was, if you could non-covalently attach dye to the self-assembled structure. Now, amine and sulfonate interactions were well reported in literature for quantification of amine. What I thought was, if I can have a dye which has sulfonate, can I use it to image peptides which have amines? Now, peptides have a lot of amines present on the surface. For example, the one at the end terminal is an amine. Then you can have lysine, arginines, which are all positively charged amines. Now, if I can use a sulfonate dye to attach to these peptides, this would be a very non-covalent interaction, so that I can label these structures and then visualize them with super resolution microscopy like STED. So with that idea in mind, we chose a standard peptide from the lab, which has either lysine or arginine. Then we do its self-assembly. So remember, in the self-assembly process, we do not do any labeling. We let the system self-assemble itself. And as expected, the amines would be present on the surface. And to this, you add around 0.5% or even less amount of the dye. Now, this is just a non-covalent addition, and you shake it up for 10 seconds. And then the labeling is ready. Now, once you have it, you can visualize them with confocal, which has a low resolution. But then you go to STED and you start to see much finer details. And then if you further corroborate it with TEM, you start to get the whole picture of the self-assembly process and its hierarchical organization. So indeed, we found it so. So initially, when you do a confocal imaging, you can see one-dimensional fibers, but they're not well resolved. But when you switch over to STED mode, it is the same microscope. You just switch over the mode from confocal to STED, you start to see much finer details. As you can see, especially in this slide here, in this part of the slide, where you see the blurry image on top starts to become much more fine features at the bottom. Now, to further quantify, what we did is we drew a line and found the intensity profile along this part. And what you see is with confocal, you see as a single fiber here. But as you switch to STED, you start to resolve into two independent fibers, which is indeed showing the power of super resolution imaging in these systems. And now you can actually start to image some of the fibers that we imaged were as thin as like 50 to 60 nanometers, which is way beyond the diffraction limit which confocal poses. So interestingly, we could visualize with much higher resolution. Further, when you look into TEM, you start to see that these fibers are also further made up of bundles of individual thin fibers, which are around 4 to 5 nanometers. So combining TEM, confocal, and STED, you start to get an overall picture that you had individual fibers, which are around 4 nanometers, which bundles up into 5 to 10 of them bundles up to form smaller bundles, and which will further bundle up to form larger aggregates, which you can see with confocal. So it starts to give you a very interesting insight into their hierarchical organization. Now, again, remember that in this process, we did not covalently label the dye. We just added a dye after the self-assembly was over. So to confirm that it's indeed electrostatic interaction, which is resulting in such dye labeling, we did zeta potential measurement. And you can see as you start to add dye, the positive charge on the surface of nanostructure starts to decrease, suggesting that it's indeed the dye is neutralizing the surface of these self-assembled peptides. But then it does not affect the supramolecular structure of the peptide. As you can see from here, the beta sheet formation of the peptide is not affected, suggesting very well that you're not affecting the peptide self-assembly, but you're electrostatically neutralizing so that there is a electrostatic interaction enabled dye labeling. And we did not stop with just one peptide. We did a range of peptides with lysine or arginine, also peptides functionalized on the gold nanoparticles. All of them have a positive charge on the surface, and you see that all of them can be imaged with STED microscopy with very high resolution. However, if the charge on the surface is not sufficient enough, so we found that anything below 10 millivolt of charge and negatively charged surfaces cannot be imaged. So in a way, it gives you a design rule as to if you have sufficiently charged surface, they can be visualized with confocal and with STED based super resolution microscopy just by adding these dye and shaking it up for a few seconds. Finally, the idea was to image structures in real time in a video. So what we did is, one of the questions that we always have is, if you take peptide which self-assembles and you add a protease, the protease would start to hydrolyze these peptides, and that would lead to disassembly. The question always is whether these enzymes act on the peptides, on the individual peptide molecules, or they can act on the nanofibers. And this question has always been difficult to answer in our field. So what I did is I was thinking if I can directly visualize these fibers as they are being chewed up by the protease, can we confirm what is happening? And you can see here that this is a snapshot of the video, and you can see that the fiber starts to now break at somewhere in the middle of the fiber itself, which suggests that indeed enzyme is actually acting on can also act on the peptide surface. It still doesn't rule out that it doesn't act on the monomers. It must be acting on the monomers, but it can also act on the peptide self-assembled structure on the nanofiber itself to chew it up from the middle. which was a very good insight into the self-assembly process. So now you can also start to visualize dynamic processes in solution with good resolution. So with that, I hope I've shown you that self-assembled structures, the soft matter or the nanomaterials can have lifelike function of temporal control over its structure and its function. And finally, once these structures form and degrade, you can also visualize them with super resolution imaging in solution. Here I would switch gear into what I plan to do at IBEC. This would still be a general idea about what I want to do because I do not have the results yet. So remember, I started with these two pictures to show that you have a static material versus the dynamic out of equilibrium structures that is shown here. Now, in one case, this is at equilibrium. It does not adapt to its surroundings. It does not require any chemical energy. The other one is at out of equilibrium. It's adaptive. It consumes chemical energy. Now, why I'm talking about it is, now, if you think about many nanomaterials that we use to interface with biology, what we think of is that if you have to interface with a cell, the material should have the mechanical property and the chemical property very much close to what biology has. So we make our drug delivery vehicle or our biointerface, which is made up of biomolecules like peptides or DNAs, lipids, etc., so that it matches the chemical functionality of the cell. The second thing which we make sure is that it is soft enough to have similar mechanical property as that of cell for better interaction. But one aspect that we always miss is the fact that these cells are at out of equilibrium. So they consume chemical energy, and their structures are constantly forming and breaking, including their receptors that change their position, and they form and break down as well. Wherein the biointerface or the nanomaterial that we use to interface are at thermodynamic equilibrium. So once they form, they do not break down or reform itself or adapt to changes that's happening on the cell surface. So what we need is a drug delivery system which can adapt to changes happening on the cell surface. And therefore, this idea of out of equilibrium drug delivery vehicle. So then what I propose is, as these receptors on the cell surface dynamically reconfigure itself, can we also have the ligands present on the drug delivery vesicles reconfigure itself dynamically to respond to the changes happening on the cell surface? And this will happen by using the same design what biology uses of a chemical fuel-driven system. So idea is that the self-assembled structure can only be maintained by constant supply of a chemical fuel. It's in some form a metabolic version of supramolecular system. So you have a anabolic reaction resulting in formation of structure and a constant catabolic reaction resulting in hydrolysis of structure. So therefore, the structure can be maintained only by constant supply of fuel. So now you can imagine that to keep a self-assembled structure like a vesicle, you need to constantly supply fuel, which would control the dynamicity of these ligands present on the surface. And that can be used then to target various drug delivery, various disease cells like cancer cells, and show that this has an efficient, this is the most efficient way to deliver drug into the disease state. So with that, I would not show the results here and conclude by saying that I've shown you that dynamic self-assembly is important, and taking inspiration from nature to develop materials with lifelike function can have a lot of applications in the future. And one of the way I want to use it is in creating adaptive biointerfaces for targeted drug delivery, which will be what I will work in molecular bionics lab in IBEC. So with that, I would like to thank my supervisors, both at the postdoctoral and PhD time, and my collaborators through my research, and the funding agencies in the past and the current funding agency from the Kasha Foundation and European Union. And I would thank you all, and I'll be happy to take any questions.